This is the lecture for Lab 1, which is about diffusion. So your tasks for this lab are to do 3 of the virtual lab simulations, and they all have to do with diffusion and thinking about what affects the rate of diffusion. You should read the Lab 1 instructions, and there's a video of us doing the experiment, and there's a post-lab assignment as usual, and then this lab has a lab report that is due in 1 week. So let me get you started with diffusion. So we're talking about diffusion because it's really important for moving molecules across the plasma membrane of cells. And this slide shows you that no matter what type of cell we're looking at, whether it's an epithelial cell in the skin or a neuron in the brain, all of the cells have a plasma membrane that surrounds them. The plasma membrane is made up of this molecule called a phospholipid, shown here. The phospholipid has 2 main components. It has these long tails that are hydrophobic. It's all carbons and hydrogens, so they're a type of lipid or fat. And then the head is hydrophilic, which means that it can form hydrogen bonds with water, and it is polar. You can see that there are some charges here, and also some oxygen, nitrogen, and so on, which are going to pull electrons towards them and create this polar region. So that's known as the head region. So this molecule has a dual nature. It's known as being amphiphilic because part of it is hydrophobic and will interact with fats and is repelled by water. And then part of it is hydrophilic, which means that it can easily interact with water. Let's look at this comic here. So there is a bear that looks like it's drowning, saying, "Help, help, I'm dissolving." And the bear on shore says, "But bears are insoluble." Well, that's easy for you to say. You're not polar. Get it? Polar, hydrophilic, dissolves in water. All right, tough crowd. So the plasma membrane with all those phospholipids is organized into a lipid bilayer, which results in this hydrophobic region in the core. And this is going to prevent some molecules from just simply diffusing across the plasma membrane. The hydrophilic heads face the water that's outside of a cell and the water in the cytoplasm inside. Okay, so as I mentioned, only nonpolar small molecules are going to be able to diffuse across that hydrophobic core of the plasma membrane. So these are molecules like oxygen, carbon dioxide, lipids like steroids, and fatty acids. So those are the main molecules that can just simply diffuse across the plasma membrane because they're nonpolar. Other molecules that are polar, or charged, like ions, like potassium or sodium, they'll have to use an ion channel to get across, or if we're talking about an uncharged molecule like glucose, it uses a transporter. So these are membrane proteins that are span the entire length of the membrane, and they allow passage through them to the other side of the membrane. And so this is known as facilitated diffusion. So actually ion channels are a type of facilitated diffusion, as are transporters. But you can see in this case that the molecules are still moving from high concentration to low concentration across the membrane. So all of these are examples of passive transport. Okay, so passive transport is simple diffusion and facilitated diffusion. And with facilitated diffusion, it can be either an ion channel or a water channel, or a transporter, like the glucose transporter. If you have molecules that are going in the opposite direction, from low to high concentration, then that requires energy. For example, it could come from ATP or from other sources, and that's known as active transport. Molecules that are in a liquid are always moving around. They have kinetic energy, and they move around randomly. So each individual molecule shown on this diagram here might be, going in whichever directions, kind of moving around randomly. But overall, when you're looking at a group of molecules, there's always a net movement from high to low concentration, as shown here. That difference in concentration is known as the concentration gradient. And as we saw in the last slide, that if molecules are going against the gradient from low to high concentration, then it's active transport and it requires energy. So let's look at this example here. So on the left side, we see some gray molecules and red molecules, and they're separated by something. Let's say it's a membrane that is impermeable. So like maybe these are ions, and there's channels that allow the ions through, but right now the channels are closed. And so if we look at the concentration gradient, we see that there's more gray on the right side and less on the left. and more of the red molecule on the left than on the right. So both of them have a concentration gradient. And then let's say that we open up those channels, and so now the molecules can move through this membrane. It's permeable. And so what's going to happen is that the red molecules are going to have a net movement from left to right. Again, an individual molecule can move in any direction it wants to. Like this red one here, it looks like it's actually going the opposite direction of the net movement. But overall, the net movement is from left to right for that red molecule, and eventually it will equalize across the membrane. And then the gray molecule goes the opposite direction. So each molecule follows its own concentration gradient, regardless of what the other molecules around it are doing. So I have a question here. What would happen if the membrane were only permeable to the gray solute, so the gray molecule? Well, we would see that the gray one would go from the right side to the left side, so it would equalize across that membrane, and then the red one would just stay on the left side because it cannot get across the membrane. So what affects the rate of diffusion? So this is an experiment. It's actually pretty similar to some experiments that you'll do in the virtual labs for Lab 1. So this is an agar plate. Agar's kind of like a gel-like substance, and they have put potassium iodide on one side, so this is a dye, so we'll call it Ki here, and then they put Congo red, which is a red dye, here. And then you wait 90 minutes, the molecules are going to diffuse from high concentration, right, where you've put them, to low concentration, because they're basically absent in the rest of the agar. And so after 90 minutes, we can see that the Ki molecule has moved further out, right? So it's diffused more than the red dye has. So what's the difference here? Why did the Ki dye diffuse farther in this experiment than the red one? And there's actually a couple of answers. So you may say, oh, well, maybe it has to do with the concentration. Well, let's say that these are at the exact same concentration. So what else could it be? Well, let's see on the next slide. So these are some things that can affect the rate of diffusion. So the lipid solubility, so if we're talking about a plasma membrane, if it is very, very nonpolar molecule, it'll be able to get across better than a molecule that perhaps is a little bit more on the polar side. And of course, if it's polar, like all the way polar, then it can't diffuse across at all. The size of the molecule is another thing, right? So larger molecules can't get across the membrane. The concentration gradient, so the difference in concentration, the membrane surface area, so if you have more membrane here, then it's more likely that the molecule will kind of randomly hit the membrane and go through. The composition of the lipid, so that can affect the permeability. So there's not just phospholipids in there, there's other types of lipids as well. Temperature affects it too. So there's all sorts of different parameters that can affect the rate of diffusion of these molecules. In that last experiment, the difference was the molecular size of the molecules. So the molecule that moved the furthest was a smaller molecule. So you'll explore some more of these in the virtual lab simulations. Okay, so why are we doing a whole lab just on diffusion in our human physiology lab course? Right? What does this have to do with physiology? Well, it turns out that diffusion is really important. As I mentioned earlier, we have molecules moving across membranes all the time. So for example, the diffusion of oxygen and carbon dioxide in the lungs. That's all about diffusion, right? So we see oxygen moving from the airspace across a couple of plasma membranes and then into a red blood cell. We also have a lot of diffusion occurring in the digestive tract. So in the intestines, like say we just ate these sugar molecules, we need to absorb them across these cells here in the outer layer and then into the capillaries. That all is diffusion across membranes. And then also in the kidneys too, this is a tube that's found in the kidneys. There's a bunch of copies of these. It's called a nephron. And blood goes in to the tube, and then we selectively bring sodium and water and other molecules back into the blood. So it's a way of regulating the concentration of these molecules in the blood, and everything that we don't bring back in is released as urine out of the body. And so this is a lot of diffusion between the blood and the kidney tubes. Okay, so what's the main experiment that we're going to be doing in Lab 1? So we're going to be modeling the process of diffusion across a selective membrane by using dialysis tubing. So dialysis tubing is semi-permeable. It's made out of cellulose, so some molecules can go through it and others cannot. So it's kind of like a plasma membrane. And so we're going to try and figure out what the tubing is permeable to. I want to just briefly mention what dialysis tubing is. So it's used in the process of dialysis, which is when someone's kidneys are no longer functioning and maybe they're waiting for a kidney transplant, then they need to be put on dialysis. And it's a way of kind of artificially filtering the blood to get rid of excess ions, salts, water, waste products, etc. So basically someone will have dialysis done for a couple of hours, and the blood, it exits their body through this tube, and then it goes through this device, and that's where the dialysis tubing is located. So throughout this device here, this tubing is found, so that's our semi-permeable membrane. And so we have the blood from the patient, and then on the other side you can control whatever solution you want here. You can have different salt concentrations or sugar or whatever. And so then the molecules that you don't want will move from the blood through the dialysis tubing into this kind of waste solution, and then that gets removed out of the machine, and then everything that's left in the blood will go back into the person here, right? So it's kind of acting like the kidney. So we need the dialysis tubing to be semi-permeable. We don't want to lose everything. We don't, want to make sure our proteins and red blood cells and so on stay inside the blood, but we want to be able to control the concentrations of smaller molecules. So here's the setup for the experiment. We're going to have a beaker, and it's filled with water and iodine, potassium iodide, which is a yellow substance. So that's why it looks yellow here. And then inside of the dialysis tubing here, we put starch and glucose, which is kind of a white-looking solution. And so what is starch? Starch is a polysaccharide that is made out of glucose, right? So it's just a bunch of glucose molecules attached together. And so what we might expect is that some of these molecules are going to diffuse between the bag and the beaker, right? And we've created these concentration gradients where there's a lot of glucose inside of the bag, no glucose outside of the bag. And so if glucose is, or if the bag is permeable to glucose, it should go from the bag into the beaker until it's equalized, right? And same with starch. The I2Ki, there's more of it in the beaker solution. And so if the bag is permeable to I2Ki, then it's going to go into the bag. Does that make sense? So once this is all set up, then we wait 35 minutes or so to allow diffusion to occur. So how will we know where the molecules go, right? So how do we know if starch has diffused through the dialysis tubing or dialysis bag? So there are two main tests that we're going to do, and they both involve color changes. So the first one is with starch and iodine. So starch on its own, I mentioned, is white in color, kind of like a cloudy white. The I2Ki is yellow on its own. However, if they come in contact with each other, they react and they create this blue or kind of like black color. And so if the I2Ki can diffuse into the dialysis bag, then we would expect the inside to change color to like blue or black. If starch is able to diffuse through the dialysis tubing, then we would expect that it would come out and go into the beaker where I2Ki is, and that we would see the beaker solution change color, right? And if neither of them go anywhere, then there's not going to be that color change. Okay, and then for the glucose, we have to do another test known as the Benedict's test. So what we do in this experiment is we have 3 test tubes, and we fill the test tubes with liquid from the dialysis bag or beaker after we've waited 30 minutes for diffusion. And so let's say in this first one, we take solution from the beaker. Okay, so beaker solution. Just fill it up into that tube, and then from the bag, we'll take another solution and put it into that middle tube. And then as a control, we're going to put water in this one. So think about whether this is a negative or a positive control. So a negative control means that we would expect nothing to happen, right? We would expect not to get a color change. If it's a positive control, then that would be a solution that has glucose in it, and so we definitely will get the color change, right? And so this is our negative control, because we expect nothing to occur here, because it's just water. There's definitely no glucose in there. Okay, then we add this thing known as the Benedict's reagent. So Benedict's reagent, which is blue in color. You'll see that in the video. Okay, so it has copper in it. It doesn't really matter, but that's why it's blue. And then we need to just heat that up to get this chemical reaction to occur. So we do that for 5 minutes. Put it in a water bath for 5 minutes at a high temperature. And if there's glucose present, then it will react with the copper and basically reduce it. And then that will cause this copper ion to precipitate out of solution. And that's the color that you actually see. Okay, so remember that the Benedict's reagent on its own was blue, right? So these tubes are going to be mostly blue. You'll see in the video that one of the tubes is not actually purely blue because there was this color change that we see with the starch and I2KI. So just be aware of that. So if there is glucose present, then we expect a color change, and it depends on how much glucose there is. So it could look green or maybe orange, or if there's just a ton of glucose, then it might look red. And so that's actually what we're seeing in this picture here. This is in the presence of glucose, right? And then the one on the left, the blue one, is just like a negative control. Okay, so does that make sense? So you should look at the video, which shows you these tubes before we put it into the heat, so you can see what the color is. And then when we heat it up for 5 minutes, then you can look at if any of them change their color. We expect at least one of them will, because we did add glucose to the dialysis bag. Before you watch the video of the experiment, I suggest that you make a prediction. Which molecules do you think will go through the membrane? So your choices are the I2KI, the glucose, or the starch. So what combination of molecules will go through there? And I'm just adding water here just to mention that there is water in both the beaker and the dialysis bag. We can't really tell which direction it's going from our setup. I'll just tell you that the dialysis bag is definitely permeable to water, so that's moving as well. But there's no test to actually see where it goes. So based on your prediction of which molecules will go through, then you can try to determine the color changes and Benedict test results that you expect. This is a really good exercise to do before you actually look at the results. So please do that. So now that you have your prediction, you can write a hypothesis. So the hypothesis should be concise but complete. So this could be 1 or 2 sentences. And you'll talk about the results that you expect, and then also why you expect those results. So if you think I2KI is the only molecule that's going to be going through that membrane, why is that? What makes it special? Is it based on the size of the molecules? Is it based on the polarity or something else? So you need to have some explanation as well. And one important note here is that your hypothesis does not have to be correct, but it should be logical. So it should make scientific, logical sense. So for your lab 1 hypothesis, you should have the following in it. So what is the bag permeable to? What's the rule for what goes through the bag and what does not go through the bag? So as I said, maybe it's based on size of the molecule, maybe it's based on polarity, or maybe it's based on something else. What color do you expect the solutions to be after 45 minutes? So thinking about the starch and I2KI part of the experiment. And what results do you expect from the Benedict's test? And like in terms of color changes. So you need to actually be specific about the colors that you expect to see. And then you'll write this hypothesis based on the predictions that you just made. And this will be an answer to one of the post-lab assignments, but it will also be in your lab report as well. And again, the hypothesis does not have to be correct, although you are seeing the results before you actually write your lab report. So I suspect most of your hypotheses will be correct. Just make sure that it's concise and complete.